Good morning, studentistas, and welcome back to Diana Knows. Hi, I am Diana, and today we will be talking about DNA, codons, and anticodons. First, let's talk about DNA. DNA stands for deoxyribonucleic acid. DNA is present in all living organisms such as animals, plants, bacteria, and humans. Even though all of the said organism has DNA, the composition of DNA found in each organism differentiates them from others. Let's now focus in humans. Human body is made up of billions and trillions of cells, and almost all cells have DNA in their nucleus. The composition of a person's DNA is 99% the same with another person, and the remaining 1% of the DNA is the only one that makes one person unique. So the question now is, what is DNA and why is it important? DNA is necessary because it contains hereditary information such as color of the hair, eyes, and other characteristics that can be seen physically, as well as diseases that is passed from generation to generation. We will now start to talk about DNA in a more detailed discussion. DNA function is to synthesize RNA by a process called transcription that is further translated into proteins by a process termed as translation. Both processes will be further discussed as we go on into the discussion. DNA is a double-stranded structure that is composed of chemical compounds which are made of phosphoric acid, deoxyribosugar, and nitrogenous bases. There are two types of nitrogenous bases. One is purine, which is composed of adenine and guanine. Two is pyrimidine, which is composed of thymine and cytosine. Each strand of DNA is made up of alternating phosphoric acid and deoxyribosugar. Moreover, nitrogenous bases is attached in between the strands of alternating phosphoric acid and deoxyribosugar. Moving on, the combination of a phosphoric acid and deoxyribosugar and one nitrogenous base is called nucleotide, which is bounded together by hydrogen bonds which are weak in the middle. That's why DNA is easily split. DNA is shaped like a ladder that has been twisted, in which the side rails pertain to the double helix structure made of sugar phosphate, and the steps are made of 4 bases. Adenine is A, thymine is T, guanine is G, and cytosine is C. Remember two things. 1. Purine bonds with specific pyrimidine, in which adenine binds with thymine and cytosine binds with guanine. Number 2 is that the importance of DNA lies on its ability to control the formation of proteins.
So this is the mRNA and this is the codons that are popping out. Then we also require ribosomes, which will help in the protein synthesis. And the third thing which connects the ribosomes and mRNA together is called tRNA or transfer RNA. The structure of tRNA is like this, with different loops coming out from different regions. It has a particular site for attachment with the amino acid because the tRNA is the carrier RNA which will carry the amino acid for the particular codon it recognized. Now we go to the anticodon, which is again 3 nucleotide stretch sequence that is found in a specific loop present in tRNA. So if you look at the tRNA structure, the main thing here is the loop in the bottom. So this loop is called anticodon loop because this loop contains 3 nucleotide sequences which are termed as anticodon, and the 3 nucleotide sequences that are found in the mRNA are termed as codon. The pairing works like this. For example, we have UUU as anticodon found in the tRNA. So the complementary codon should have AAA because remember the number 1 in the two things that I told you to remember. Purine binds with specific pyrimidine, so A binds with U and G binds with C. This anticodon UUU will be attached to a specific amino acid, where in this case is phenylalanine, because UUU attaches to phenylalanine. So you can see that the tRNA is the transfer RNA which is responsible for bringing all the amino acid but they are very specific for a particular type of amino acid only.
And now let us move on to codons and anticodons. Codons, which are found in mRNA, are the complementary triplets of the code triplets of DNA genes, while anticodons are found in tRNA that is complementary to codon. These two terms are related to each other. Codon is a 3-nucleotide stretch of sequence which codes for proteins. Looking back at the central dogma of biology, this begins with a DNA molecule. From the DNA, it produces mRNA, and from mRNA, it produces protein. mRNA, which have different bases coming out because it is a single-stranded structure, and 3 of those bases coming out are called codons, which are specific for coding a particular amino acid. In mRNA, there are many types of codons, which is placed in a chart called genetic code. When you look at the genetic code, you will find many different codons as well as all different amino acids that those codons are coding for. Codon means 3 nucleotide in which each of the 3 nucleotide is specific for coding a particular amino acid. Now, to interact with those 3 codons, we need opposite 3 nucleotide sequence which will sit onto the codon and read that codon, then bring out the right amino acid. Because during the protein synthesis, we require 3 major molecules. One is the mRNA, which will be coded into proteins. To make it more simple, we have this strand of DNA that will move from 5 to 3. So for the complementary strand, you will have A paired with T, T paired with A, and G paired with C, and so on and so forth. Messenger RNA is similar to DNA, but instead of thymine, you have uracil. So now for this complementary strand, the DNA will split into 2 by the polymerase enzyme. Then there will be single DNA strand that is left, and eventually, the T of single strand will pair with the A, and A with the U, and C with the G, and so on and so forth. Now, messenger RNA pairs with the transfer RNA. The messenger RNA will be the codon, and the transport RNA will be the anticodon. The transfer RNA has uracil, therefore A will pair with U will pair with A, and G with C, and then so on. Now, let's find the amino acid by using an mRNA chart. So I will use the messenger RNA codon. mRNA will code in triplets. AUG, which is a start codon, pairs up with methionine. Then I have CCU, and it pairs up with proline. Then CUA pairs up with leucine. And lastly, GUA pairs up with valine.
These 4 products are the amino acids. So here is the completed project from DNA to mRNA to tRNA to the amino acids. And that will be all for the discussion of DNA, codons, and anticodons. Thank you very much.